Good evening all. John Minor, and I'll be taking you through the unit Introduction to Zoology part the cell. So cell is the basic unit of life, and cell can be basically classified based on the presence or absence of membrane-bound nucleus. And in that case we have prokaryotic cell and eukaryotic cell. Prokaryotic cell is that type of cell that lack a membrane-bound nucleus, and it can be further classified to bacteria and archaea. In bacteria, these are the everyday microbes, whereas the archaea, these are the ones that are found in hostile environment. On the part of eukaryotic cells, there are type of cells that have a membrane-bound nucleus. So we come to the differences between prokaryotic and eukaryotic cells. Prokaryotic cells are smaller in size, usually 5 micrometer. Prokaryotic cells, they are smaller in size, usually 5 micrometer. Eukaryotic, they are larger, usually 10 micrometers. Prokaryotic lack a membrane-bound nucleus, whereas eukaryotic has a membrane-bound nucleus. DNA in prokaryotic cells is circular without proteins. DNA in eukaryotic cells is linear and associated with proteins. Prokaryotic cells lack cytoskeleton. Eukaryotic cells has cytoskeleton. Ribosomes in prokaryotic cells are smaller, whereas in eukaryotic cells they are larger. Cell division in prokaryotic cells is via binary fission, whereas in eukaryotic cells is via mitosis and meiosis. The reproduction in eukaryotic cells is asexual. Reproduction in eukaryotic cells is via asexual and sexual. Despite of the structural differences, both type of cells have some similarities needed to support life, such as they both use enzyme to regulate chemical reaction in the cell. They both carry out essential processes such as metabolism and growth. They both contain DNA that contain genetic information. So we go to the cell structure. Therefore, we will find the cell organelles such as cell membrane, cytoplasm, mitochondria, endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, and centrioles. We start with cytoplasm, which is a semi-fluid matrix containing organelles. Its function, it is a site for metabolic reactions. We go to mitochondria. It is a bean-shaped organelle with double membrane, the outer and the inner membrane, which is greatly folded into crystal. Its function, the site for cellular respiration, and produces the ATP, adenosine triphosphate energy. We go to endoplasmic reticulum. We have rough endoplasmic reticulum and a small endoplasmic reticulum. In rough endoplasmic reticulum, it is involved in protein synthesis and transport, whereas the small endoplasmic reticulum is involved in lipid synthesis and detoxification. We have the other organelle as ribosomes. They are found free in cytoplasm or attached to the rough endoplasmic reticulum. They are site for protein synthesis. We have the other organelle as Golgi apparatus. They are stuck of flattened membrane sacs. Function, they form vesicles for secretion, modifies, packages, and transport proteins and lipids. Go to the other organelle, lysosome. They are small. Their function is to break down old and unwanted cell organelles via the hydrolytic enzymes. When we go to centrioles, they play a role. They play a role in formation of spindle fibers during cell division. Let's go to the last one, cell membrane. It is a thin, flexible layer that surrounds the cell. Its main function is to control materials that enters and leave the cell. In short, selective movement of substances in and out of the cell. It contains a phospholipid bilayer. Each phospholipid layer has a hydrophilic head that attracts water and a hydrophobic tail that repels water. These arrangement forms a barrier that prevents unwanted substances from passing freely. They also have an embedded layer of protein which perform important functions such as transporting substances and acting as receptors. Thank you.
So, my name is Derek Karyoki. I'm in group number 34 and I'm taking you through evolution. So, what is evolution? Evolution is the gradual change in genetic makeup of a population over generations, leading to differences in traits and sometimes the formation of new species, sometimes lead to formation of new species. So there are two types of evolution, that's microevolution and macroevolution. So what is macroevolution? Macroevolution refers to the small scale changes within species over a short period of time. It occurs over a short period of time. And the example includes the bacteria becoming resistant to diseases. The bacteria becoming diseases shows the microevolution. So after there, I'm going to move to the agents of microevolution. So there are several agents of microevolution, which include the gene flow, the mutation, the genetic drift, natural selection, and random mating, and sexual recombination. I'm going to explain one by one as for more understanding. Okay. So I'm going to start with mutation. So mutation here, this occurs within genes of organisms in specific species. When there is a changes in genes or the genetic material, it leads to formation of new species, inhibiting or showing the agent of microevolution. I'm going to move to gene flow, which is the movement of individuals of the gametes between populations. The gene flow also the migration shows the movement of organisms from one part to another, or movement of gametes between population. Movement of gametes between population will lead to formation of new species over a short period of time, showing microevolution. So genetic drift. It is the random change in allele frequencies due to a chance. It has a stronger effect in small population and can lead to the loss of alleles. One allele becomes 100%. So I'm going to move to natural selections. The natural selection. So natural selection favors individuals with the advantageous traits. In short, the individuals that will be able to survive a specific environmental condition is going to survive in that condition, showing microevolution. So I'm going to move to random mating, which occurs in individuals to mate based on specific traits. In short, on random mating, individuals choose mates to or with specific traits. That's showing microevolution. So I'm going to move the continental drift, movement of continents or environment. This occurs drift refers to the movement of continents. And when this continent moves from, for example, from one place to another, some of the species will be moved to that place, showing microevolution. So I'm going to move to adaptive variation, which is the evolutionary process in which descendant populations spread from ancestral group into other areas. So adaptive variation occurs to several factors, which were natural selection, changes in physical and biological environment, and changes in the levels of oxygen leading to adaptive radiation. Next, I'm going to move to macroevolution. So, what is macroevolution? So, macroevolution refers to large-scale evolution changes that occur over a long period of time. It is a large scale which differentiates it from the microevolution. On macroevolution, I'm going to explain the causes of extinction. So we have several causes that cause extinction of new species, which included we have the climatic change, we have volcanic activity, collision of asteroids, continental drift. I have many others that are not treated, which include the human activities. So I'm going to start with the climatic change. So, in a given environment, when the climate change, it can favor or lead to extinction of a given organism. For example, we have those organisms living in deserts. When the climatic will change, that climate will not favor those organisms, leading to the extinction of those organisms. We have volcanic activity. Volcanic activity or volcanicity may lead to destruction of a specific species. We have collision of asteroids, for example, asteroids from space may land. For example, that one that destroyed the old generation of dinosaurs may lead to extinction of organisms. We have continental drift that I talked about, the movement of continents, leading to extinction of organisms from another continent. We have human activities, for example, the tree cutting, which may lead to extinction of organisms. So that will be the end of the topic. Thank you.
So hello, my name is Leila Chimoni. Group 34. So my topic is resistance to diseases. I've written there. And so when we talk about resistance to diseases, we are simply talking about the body's defense system. And the body's defense system is composed of the immunity and immune system. So the immunity refers to the body's ability to resist all types of organisms or toxins that damage the tissue or the organs. And then the immunity systems, it just serves to defend the body from microorganisms. So the body's defense system is comprised of two components. We have the specific and non-specific defense mechanisms. So this specific is also called innate immunity. But this one is called acquired or adaptive, the specific one. So we'll start with non-specific. And here in non-specific, this is a type of defense system that offers immediate protection from microorganisms and other foreign particles. And here we can talk of 6 aspects. We have the skin, body secretions, phagocytosis, neutrophil killer cells, inflammation, and proteins. So let's start with the skin. Here, the skin, we can talk about a few things. We have the epidermis. Epidermis has a tough keratin layer that can't be easily digested by microorganisms. The second thing, we have the under skin, we have sweat. The skin produces sweat, and sweat is acidic, therefore inhibiting growth of pathogens. And then we have the sebaceous glands. They produce sebum, and sebum is antibacterial, meaning that it prevents bacteria from entering the skin. And then we have commensal flora. Commensal flora, they just compete with pathogens that inhibit, therefore inhibiting their growth. And then lastly, about the skin is that it is constantly renewed, therefore repairing any damage, forming a renewed ban against infection. Secondly, we have body, so we're done with the skin, we have body secretions. Under body secretions, we talk of mucus, tears, saliva, earwax, and also the cilia. So mucus, it covers body membranous body surfaces, and therefore keeping them moist. And then it also lines the nasal cavity, digestive tract, vagina, and uterus. And then you have in the respiratory tract, you have the cilia. Cilia traps microorganisms, and it suits them into the pharynx. From the pharynx, they are taken to the stomach, and they encounter mucus, acids, and enzymes. And then the fourth one, we have ears, which produce wax, or it's also called cerumen, and wax traps foreign particles. And then saliva and tears produce lysozyme enzyme, which lyses bacterial cells. And then we have the phagocytosis. So phagocytosis refers to the engulfing of pathogens. And here we have phagocytes. These are white blood cells that destroy pathogens and dead tissue. And there are neutrophils, macrophages, and monocytes. Yeah. And then we have neutral killer cells. Neutral killer cells, these are a special population of white blood cells. And their function is to destroy cancer cells and cells infected by viruses. And then we have the inflammation. Inflammation refers to a local reaction to tissue damage or injury. So its characteristic is swelling, redness, pain, and heat. And its function, one is to help fight infection, and the second one is to heal the wound. Finally, we have the proteins. Proteins here, we can talk of three types. The proteins that take part in non-specific defense mechanisms. We have complements, interferons, and interleukins. Complements, these proteins are produced by the liver, and their function is to lyse bacteria and attract phagocytes to infection cells. We also have the second one is interferons. Interferons, they are produced by virus-infected cells, and their function is to interfere with the replication of viruses. And then we have interleukins. Interleukins, these are proteins that promote inflammation. That's the function. So we are done with non-specific defense mechanisms. Let's go to specific. Specific defense mechanisms. Their primary function is to discriminate between self and non-self. And here we can be divided into two. We have humoral response and cellular response. And the humoral response, the top of them, it's mediated by B lymphocytes here. And they are produced by antibodies. Those antibodies are found in the serum, and then their function is, its function it is used against bacteria and viruses that have not invaded cells. And then we have cellular response. We have cellular response. This is based on productions of cells that are sensitive to the cell, and it's mediated by T lymphocytes. So that's all about specific defense mechanism or acquired immunity. So that's all about resistance to disease. Thank you.
Hello, I'm David Kilango, group number
Part 34, that will be taking you through kingdoms of life. Specifically, we'll be taking you through kingdom Protista. So the kingdoms of life we have kingdom Protista, kingdom Antibacteria, the bacteria, Animalia, Plantae, and Fungi. So I will be taking you through kingdom Protista. So the general characteristics of kingdom Protista is that they are unicellular organisms, Protista, and unicellular, for example, algae, and they have biologic cells, and movement is by use of structures such as cilia, flagella, and pseudopodia, and the production is both sexually and asexually. So it has these phylums. Kingdom Protista has these phylums. You have phylum Sarcodina, phylum Granulosa, phylum Actinopoda, phylum Pseudopoda, phylum Anticomplexa, and phylum Rhinozoa. So you can give phylum Sarcodina. And phylum Sarcodina, example of an organism in phylum Sarcodina is amoeba. So the characteristics of organism in phylum Sarcodina is that they are unicellular, eukaryotic organism, and their bodies they have a definite shape, they have a regular shape, and movement is the use of pseudopodia, which are phosphate, and reproduction is asexually through binary fission, and they mostly inhabit aquatic environments, and their method of feeding is by phagocytosis, whereby they engulf their food particles and they form food vacuoles. They inhabit mostly freshwater and aquatic environments. The second one is phylum Granulosa. Example of an organism in phylum Granulosa is Globigerina. The characteristics of organisms in this phylum is that they are mostly marine and their bodies are covered with a calcareous shell, and movement is by use of the pseudopodia, which are fine branching pseudopodia phylum networks, and the production is both sexual and asexual. And nutrition is that they feed on small organisms and small particles, organic particles, and their shell is multi-chamber. They have a special feature whereby their shells accumulate to form sediments, for example, limestone and chalk. The third one is phylum Actinopoda. Example of an organism in phylum Actinopoda is Actinophilus. So the characteristics of organisms in this phylum is that they are spherical unicellular organisms, and locomotion is the use of axopodia, and they also use this axopodia for feeding. And they have the reproduction is asexually through binary fission, and the habitat is mostly freshwater and marine. The fourth one is phylum Pseudophora. Phylum Pseudophora example is Paramecium. So the characteristics of organisms in this phylum is that their bodies are covered with cilia, which are short hair-like structures, and movement is by coordinated beating of this cilia. And they exhibit nuclear dimorphism, whereby they have two types of nucleus. The first one is macronucleus, which controls the metabolism, and the second one is micronucleus, which is used for reproduction. And nutrition is mesozoic, whereby they feed through an oral route. And reproduction is both sexually and asexually. In asexual reproduction, they produce by binary fission, and sexual reproduction is by conjugation. The habitat mostly is freshwater environments. The fifth one is phylum Anticomplexa. Example of an organism in phylum Anticomplexa is Plasmodium. The characteristics of this phylum is that they are all parasitic, they lack locomotory structures and other stage, and they have a unique structure called complex, which is specialized in penetrating through host cells. Their nutrition is that they absorb nutrients directly from their host. The reproduction is both sexual and asexual. And the next one is example of an organism in phylum is the gleaner. These are unicellular eukaryotic organisms, and the locomotion is by use of two or one flagella, and they show mixotrophic nutrition, whereby some are autotrophic and others are mixotrophic. The production is by asexual reproduction, whereby they exhibit longitudinal binary fission, and mostly they inhabit their freshwater ponds, but some live in marine environments. So these organisms, they are of economical importance, whereby they provide ecological balance. The other one is that they are they disable nutrients in the environment, and also they are indicators of environmental health. Some are used in sewage treatment, and also some are of medical importance, whereby we learn because some causes diseases. So we learn that we develop medicine to control these diseases. And the last one is that they are used in research and education. And thank you.
Platyhelminthes. The name plat means plat, while helminthes means worms. Therefore, platyhelminthes are animals commonly known as platforms. Their bodies are soft, thin, and flat. Some are free-living, while others are parasites. Their general characteristics are: their body is dorsoventrally flattened. This means that it's flat from bottom to top. The other characteristic is their body is bilaterally symmetrical. This means that the left and the right side are equal. The other one is their body is soft and unsegmented. The other one is gaseous exchange occurs through diffusion through body surface. And the last one is they have no circulatory system. Therefore, respiration does not occur by circulatory system. The other topic of discussion is their habitat. They are found in fresh water, marine water, and moist places. Some live inside animals and humans as parasites. Their nutrition: free-living platforms feed on small organisms. Parasitic platforms absorb food from the host body. Now the other topic of discussion is their reproduction. Most are hermaphrodites. This means they have female and male organs together. And also they can reproduce sexually, and some can reproduce asexually by regeneration. The examples of animals from phylum Platyhelminthes are: number one, planaria. These are free-living freshwater platforms, so they live in fresh water. And the other one is phylum, tapeworm, found in small, they are found in intestines, not necessarily small. Their importance: they are harmful and also they are useful. And I'll start with the harmful. Tapeworms, they cause illness in humans. And also, liver fluke affects livestock. This leads to lower production and lower yield. And then next, I'll go to the useful. The useful are the nice things. And planarium, because we said some they reproduce asexually by regeneration. Now this can be a useful thing because planaria, they are used in scientific research because of regeneration. And now I'll finish off with the adaptation. They have hooks and suckers for attachments. The other one, they have big body for coverage is for their protection. The other one, they have high egg production. And the last one, they can live or survive with little amount of oxygen. And that is the end of my discussion.